I'm Gagandeep Kang. I am a medical scientist. I trained in medicine and microbiology at the Christain Medical College in Vellore in Tamil Nadu. I finished my training and then stayed on at CMC as faculty, where I've spent essentially my entire career, now about 30 years, doing research on enteric infections in children and on vaccines, nutrition, water, and sanitation. Initially, when we started out, we also heard a lot about aerosols and what aerosols meant, and a lot of emphasis on the fact that probably we didn't need to wear masks because the droplets that SARS-CoV traveled in were really large droplets, and masks were meant for much smaller droplets. That got me to think about what are the physical determinants of risk of infection. How much do temperature and humidity matter? What kind of filtration of a mask is required? How far should you be for physical distancing to be effective? What are the changes that happen when someone speaks loudly or sings or talks? And all of these really have a mixture of the biological characteristics of the virus and the physics of how a viral particle moves in the environment. In the case of SARS-CoV-2, we have a pathogen that is new to the whole world. And when people get infected, it's the first time they are seeing this agent. So what we should be seeing is disease that is similar across the entire world. And yet that doesn't seem to be the case. In India and in Africa, we seem to see that the proportion of people who are infected but don't become sick is much larger. And among those who are infected and get ill, the severity of the illness is less. I think one of the reasons for this might be the fact that we see many more pathogens in our kinds of environment, and that may modify the response to SARS-CoV-2. What I would like to do is to try and figure out how we could measure the infection history of a person and then see whether that modifies their response to SARS-CoV-2 and explain why we are not seeing severe disease in India and in Africa. In the development of vaccines, it's very important for us to know what a vaccine does to actually protect people. It's quite surprising that for a lot of the pathogens for which we have vaccines, we actually don't know what the protective mechanism is. So for SARS-CoV-2, while we have more than 300 vaccines in development, it has been a question that do we know which kind of antibody or cell-mediated immune response really protects us from infection, what's necessary, and what is sufficient. I'd like to be able to work on defining the correlate of protection from vaccines so that when we develop vaccines in the future, we will not need to go through the kind of hundreds of thousands of people being recruited to test a vaccine that is required today in clinical trials. When we think about pandemics and infectious diseases, finding solutions is important, and those solutions really need to be science-based, whether they are measures of risk, measures of disease, or measures of protection. With the technologies that we have today, I think that it's very possible that we can do this for SARS-CoV-2.